Let's talk about what we know about the biology of depression. There are many theories. The first is the monoamine hypothesis. We know three neurotransmitters modulate mood: norepinephrine, serotonin, dopamine. They're all monoamines. Medications like SSRIs prevent the reuptake of the monoamine serotonin in the synapse between two serotonergic neurons, thus leaving that synapse aflood with serotonin. However, that's far too simplistic because we know that after a little while of excess serotonin in the synapse, there's all kinds of postsynaptic remodeling. Receptors are upregulated, downregulated. There's second messenger systems that then go down to the DNA level and modulate genetic expression of all kinds of proteins. Call that theory the synaptic remodeling. There's also the brain-derived neurotropic factor hypothesis. It turns out that when you look downstream from these receptors and second messenger systems, a neurotropic factor called brain-derived neurotropic factor is upregulated. There's increased genetic expression of this protein, and this protein is key in supporting growth of neurons and synapses. There's a neuroendocrine abnormalities are found in depression. We'll talk about how biological rhythms like sleep are affected. We'll also talk about the adrenal, the hypothalamus pituitary adrenal axis is altered in depression. And then finally, we'll talk about how depression is a disorder of inflammation. So here's the whole complicated slide about the monoamine hypothesis. It starts out with tryptophan and tyrosine, and through enzymatic cleavage, they become serotonin and norepinephrine. Then you've got enzymes to degrade these neurotransmitters. You've got release of the neurotransmitters, reuptake pumps over here, and you've got receptors in the postsynaptic neuron, and they activate second messenger systems, which then go down to the DNA level. We know that serotonin is an important neurotransmitter, not just because our drugs work for depression, but also because if we deplete serotonin in the brains of animals and even humans, you can actually precipitate depression. In humans, there's a tryptophan deficient diet that causes reduced production of serotonin, and that's been associated with depression. If we do lumbar punctures in depressed patients and look at their cerebrospinal fluid, we see reduction in a metabolite of serotonin, 5-HIAA. Turns out that platelets contain serotonin inside. Serotonin is important in platelet aggregation. We can find that there are lower concentration of serotonin uptake sites in platelets. Another finding of deficient serotonergic function in patients with depression. Finally, if you take suicide attempters and do spinal taps of those individuals, you'll also see low CSF concentrations of 5-HAA. This is also true in people who are violent, particularly if they can commit impulsive violence. We also see low serotonin metabolites in their cerebrospinal fluid. Most antidepressants increase the levels of synaptic serotonin, and most antidepressants also cause downregulation of postsynaptic serotonin receptors, and that downregulation lags a week, 2, 3 weeks, or even 4 weeks, explaining why it takes antidepressants so long to work. Well, norepinephrine is the other key monoamine, and some antidepressants like the tricyclic dizipramine are almost totally noradrenergic in function. These drugs also lead to downregulation of postsynaptic beta-adrenergic receptors, and again, that takes a few weeks, and that parallels clinical response. Antihypertensive drug reserpine depletes noradrenergic stores and can precipitate depression, hence we don't even use it anymore in most people for hypertension. The third monoamine is dopamine. The role here is less clear. Dopamine activity may be lower in depression but increased in mania. There are some antidepressants that increase levels of synaptic dopamine, bupropion being the main one, and it, via inhibiting reuptake of norepinephrine and dopamine in those neurotransmitter systems, again you get alteration in receptors and second messenger systems that lead to improvement in mood. We know that depression is common in Parkinson's disease, about 40% comorbidity, and Parkinson's, of course, is a state of dopamine depletion, again implicating dopamine in the regulation of mood. So here you've got our three neurotransmitters, dopamine, norepinephrine, and serotonin, overlapping in the regulation of mood. We see that dopamine is where we think motivation and pleasure are controlled. Norepinephrine, you've got alertness and energy. And serotonin, obsessions and compulsions seem to be very closely tied to serotonergic function. Well, glutamate is going to be the neurotransmitter of this decade. We now have found that ketamine, which is an anesthetic and recreational drug, shows immediate efficacy when given intravenously to patients with treatment-resistant depression. In fact, patients who have been suicidal all their lives, when they get IV ketamine, the suicidality disappears, at least for a week or two. Researchers are now working night and day trying to figure out how to get ketamine into an antidepressant formulation that can actually provide durable antidepressant response. Anyway, ketamine is a NMDA receptor antagonist, just like PCP. It works on blocking NMDA receptors, and that's the glutamatergic system. What model do we use to test antidepressants? Well, the most common one is the forced swim test. We take these unfortunate mice and put them in a glass column with water, and we see how long does it take for the mouse to give up. A non-depressed mouse will struggle for several minutes trying to keep its head afloat. A depressed mouse, on the other hand, will give up very quickly, will nearly drown, but the experimenters have some mercy and will take the mouse outside of the column after we've seen how long it takes for it to go under. Now, an antidepressant given to one of these depressed mice will actually result in a prolongation of the struggle to remain afloat, and that's how we can test antidepressant efficacy. Now, I want to show you just some of the key brain regions associated with depression. So first, we see the hypothalamus associated with regulating sleep and appetite. The amygdala is really where mood resides in the brain, and we'll be talking about the amygdala throughout the course. The prefrontal cortex gives us some of the cognitions associated with depression, guilt, suicidality, and worthlessness. The nucleus accumbens is the pleasure center in the brain, and a deficit over there can lead to decreased interest, decreased pleasure, lack of motivation. Let's take a look about how the neurotransmitters modify genetic expression. So let's look here. Neurotransmitters bind to a receptor. ATP comes along, gets dephosphorylated, and we get cyclic AMP. Cyclic AMP prods the protein kinase to go down to the nucleus and bind to the cyclic AMP response element binding protein. The protein kinase phosphorylates that CREB, making it into a transcription factor. It goes and binds a promoter gene right over here, which then results in transcription of an important protein. That new protein that's being transcribed then goes on and does some of the work of reconfiguring the monoaminergic synapse and architectural system. So we know patients taking antidepressants at the time of death show increased CREB levels. Unmedicated patients show decreased levels. Depressed patients are less responsive to CREB activation, and CREB activation is greater in patients who respond to therapy versus those who do not. And this occurs with psychotherapy, not just drugs. We talked a little bit earlier about brain-derived neurotropic factor. Well, it looks like CREB is directly involved in activating the genes that produce brain-derived neurotropic factor. So antidepressants increase the serotonin or norepinephrine in the synapse. They go down to the receptor, activating second messenger system. Cyclic AMP comes along, activating protein kinase A, which then phosphorylates CREB. CREB then goes down to the promoter region of the BDNF, and BDNF is transcribed. These trophic actions alter synaptic plasticity and actually promote growth of neurons and their connections. BDNF is concentrated in the hippocampus, cortex, and basal forebrain. The levels are increased by antidepressants, electric shock therapy, intellectual stimulation, exercise, caloric restriction, and even sleep deprivation. And curiously, all of these can have impacts on mood. Let's talk about another pathophysiologic anomaly seen in mood disorders. So I believe you've all learned about the hypopituitary adrenal axis. Hypothalamus produces ACTH, which stimulates the pituitary to make corticotropin-releasing factor, and that factor goes down to the adrenal gland and stimulates production of cortisol. Then you have cortisol, once produced, has a negative feedback loop which goes all the way back to the hypothalamus and says, hey, slow down all this production. The hypothalamus has reduced ACTH, the pituitary then has reduced corticotropin-releasing factor, and there's less stimulation of cortisol production by the adrenal gland. Well, what happens in depression? This negative feedback loop is defective. So when cortisol is made by the adrenal gland, rather than it going back and telling the hypothalamus, hey, shut down this system, it doesn't do that. And so you have a chronically elevated cortisol system in the brains of patients with depression. So this slide here further just illustrates what I have just told. This hypersecretion of cortisol, i.e. the lack of the negative feedback loop, is found in 50% of depressed patients. Unfortunately, it's not a laboratory test that would be all that useful since it only occurs in 50% of depressed patients. There's such a thing called a dexamethasone suppression test. So in the night, patients given dexamethasone, which is a corticosteroid, and in the morning you check their cortisol levels. Now in a healthy individual, you give them the steroid at night. In the morning, their cortisol production is suppressed, and so they have low cortisol levels. Do this in depressed patients. You give them dexamethasone at night. Guess what? In the morning, they have just as high cortisol as they would had they not been given dexamethasone. So there's a negative dexamethasone suppression test. This is not only found in depression, but in other psychiatric disorders as well. Let's talk about epigenetics. This is when environmental influences impact genetic expression, leading to a behavioral response. So I am going to show you a nice little video here about what happens when rats in utero and shortly thereafter are exposed to nurturing mothers versus non-nurturing mothers.
Choose among these mothers come from a long line of inbred rats, so their genomes are highly similar, but they care for their pups very differently. Immediately after birth, methyl molecules, green, silence the GR receptor gene, gold, in the brain cells of all rat pups. During the first week of life, the mother rat's licking and grooming releases certain brain chemicals in the pup. The brain chemicals activate cell signaling pathways that reach all the way down to the DNA. Strong nurturing signals remove methyl groups from the DNA and activate the GR gene. In pups who receive little nurturing, the signals are weak and the methyl groups stay put. Inside the brain sits a structure called the hippocampus. One job of the hippocampus is to coordinate the body's response to stress. In the hippocampus, cells produce a little or a lot of GR protein, depending on how much the pup was nurtured. During the fight or flight response, the hormone cortisol is released into the bloodstream. Cortisol binds to GR protein, causing the cell to send out calming signals. Rats who have a lot of GR protein relax quickly after stress. Rats with little GR protein remain stressed for much longer. Nurturing affects more than the GR gene. As adults, high and low nurtured rats have expression differences in hundreds of genes. In the end, what you get is two rats that behave very differently. Calm or anxious behavior can be an advantage in different situations.
So we've just seen how stressful environment leads to hypermethylation of genes that then leads to decreased transcription of the glucocorticoid receptor gene. And the fewer the glucocorticoid receptor genes that are present on the hypothalamus, for example, you're going to have a decreased feedback loop giving you the excess cortisol, and cortisol actually leads to hippocampal apoptosis. The neurons of the hippocampus, when exposed to too much cortisol, actually shrivel away and die. Postmortem studies of methylation of the glucocorticoid receptor promoter gene in suicide victims show increased DNA methylation there. Hypermethylation of not only the glucocorticoid receptor but also the serotonin transporter gene is also found in infants of depressed mothers. So stress, cortisol, and other insults decrease the DNA expression of BDNF. That decreases neurogenesis. And about 50% of patients with depression have this abnormality. We do see that hypermethylation of BDNF promoter genes occurs in many psychiatric disorders as well. So BDNF may be the common pathway by which stress decreases BDNF levels and antidepressants increase BDNF levels, leading to either proliferation of neurons or destruction. So I said earlier that hippocampal cells will atrophy when exposed to long-term hypercortisolemia. And here we look at the total hippocampal volume in 38 women, fairly young women, with depression. The greater the number of days of untreated depression, the lower the hippocampal volume. So depression actually kills the hippocampus. As I said already, that antidepressants may protect the brain via upregulating production of BDNF. Interestingly, SSRIs can improve outcome in stroke independent of the presence or absence of depression. Moving along to another pathological finding in depression is circadian dysregulation. Depressed patients don't make that growth hormone the way they should in the first third of the night. There's decreased secretion of melatonin, and there's some characteristic disturbance of sleep physiology, the most important one being decreased REM latency. In a normal sleeper, it takes about 90 minutes from sleep onset until the first REM episode. In depression, that 90 minutes becomes 30 to 60 minutes. Another very common finding in depressed patients, but again, it can't be used as a laboratory test because it's neither sensitive nor specific enough. Moving on to another finding in depression is an increase in inflammatory markers. So the increases in C-reactive protein, interleukin 1, interleukin 6, and TNF-alpha levels in depression. These inflammatory markers are reversed when the depressed individual is successfully treated with antidepressants. We know that cytokines can even cause symptoms of depression. Interferon, which causes a surge in inflammatory cytokines, causes depression in nearly 50% of patients. Okay, so let's look at this complicated slide here. You've got cerebral blood vessel leaking inflammatory markers. And you get a microglial cell. Now, what happens normally, you've got tryptophan, and tryptophan hydroxylase turns tryptophan into serotonin. That's great, that's a good neurotransmitter. However, inflammatory proteins come to the microglial cell, and they upregulate an enzyme called indoleamine dioxygenase. This IDO causes tryptophan to preferentially be converted into a toxic protein called quinolinic acid. Quinolinic acid goes to the terminals of glutamate neurons, causing excitation and ultimately apoptosis of neurons. Here you have the same illustration. Tryptophan should be going into serotonin, but you've got IDO over here that then converts the tryptophan through a bunch of different levels into quinolinic acid, which is excitotoxic to neurons. Once again, we look at stress, genetic factors, environmental factors leading to depression, leading to inflammatory cytokines and hypercortisolemia. Hypercortisolemia leads to reduced neuronal repair via reduced brain-derived neurotropic factor, and you can get shrinking of the hippocampus, which can actually lead to dementia. There is actually a relationship between midlife onset of depression and dementia. On this side, we see the pro-inflammatory cytokines, the induction of IDO, and the increase in quinolinic acid in the microglia, leading again to apoptosis of neurons. There's quite a bit of evidence that genetic factors are operating in mood disorders. It's rare to see a patient with depression that doesn't have a family history with first-degree and second-degree relatives who also suffer from mood disorders. Monozygotic twins have about a concordance rate of about 50%, and dizygotic twins less so. And this slide just illustrates what I was talking about just a moment ago about how the concordance rate in monozygotic twins is very high and a little less high in the dizygotic twins. We've talked about a lot of biological factors that underlay depression, but we also find many psychological factors. Early childhood abuse and deprivation leads to an increased risk of depression. The disruption of an attachment bond between a child and its caretaker. An individual who learns that nothing that they do ever turns out right will have a condition called learned helplessness. It's very similar to that mouse model where I showed you it fighting to stay afloat in that glass cylinder. Once it feels helpless and depressed, it will just stop struggling. Depressions also cause often anger turned inwards. There are many of cognitive distortions that we'll talk about that underlie depression, and often interpersonal difficulties can precipitate depression. Putting it all together, we have here an interaction between environment and genetic predisposition that leads to phenotypic depression. So here we've got the serotonin transporter gene polymorphism. This gene comes into, has two alleles. One is the long allele, the other is the short allele. What you see when you have homozygotes of the short allele, they're predisposed to depression. And so when given a number of stressful life events, as these increase, individuals with the short allele actually end up having a higher incidence of depression. To the contrary, the individuals with the long allele, those homozygotes, are relatively protected from depression, with the heterozygotes intermediate. You see the same thing with suicidal behavior or ideation, that the short allele version of the serotonergic transporter gene leads to increased suicidality according to the number of stressful life events. This concludes our discussion of the pathophysiology of depression.